Hello, everyone, and thanks all of you for coming to the first Heidelberg AI event this year. Just as a heads up, this event is live streamed and in person. So hello to all of you who are here online via Zoom. And please, to the people who are here, please be very quiet because we have a very sensitive microphone. And for them to have a good sound quality, it is really important that you are as quiet as possible. Now, generally, after every Heidelberg AI event, we will have some drinks where you can also talk with the speaker and socialize among each other. And I highly encourage you to do so. Heidelberg AI itself is part of an ecosystem where we have the LS Unit Heidelberg, the NCT Data Science Seminar, and we're really closely collaborating with the German Cancer Research Center because we're all affiliated with them and also Helmholtz Imaging. And all of the people who are organizing Heidelberg AI are part of the Division of Medical Imaging Computing, which is led by Professor Dr. Klaus Meyer-Hein. Now, to our speaker, Professor Charlotte Bunne. She has just been she has just started her work as the head of AI for the, I need to read it out, Artificial Intelligence in Molecular Medicine Laboratory. And she did her PhD and master's at the ETH Zurich. During this time, she won numerous awards and prizes, so many that I can't list all of them, but she basically worked at all of the important places and did research at all of the important places, so like Google, Apple, MIT, Harvard, and IBM Research. But most importantly, she actually did her bachelor's here in Heidelberg in biology, and they actually wrote her bachelor thesis at the German Cancer Research Center, which is why we're very happy to have her here. In her research nowadays, she focused a lot on solving issues in biomolecular sciences with machine learning approaches, and a lot of it with generative modeling and more specifically neural transport. And yeah, I'm very looking forward to a talk, and without further ado, I would hand it to our speaker. Please give a warm round of applause.
Yeah, so I'm going to speak about virtual cells and digital twins, AI and personalized oncology. And so actually, so I did my bachelor's here, and especially I spent a lot of time in this building, actually many days and many nights. So it's very, very nice actually being back in the seminar room in particular. So I did my PhD at ETH and at MIT, and so I was working for a year with Anne Carpenter and Chantal Singh, but my main advisors were Andreas Krause and Marco Couturi. So Marco was first at Google and then at Apple, and I had to follow. So that explains why I basically moved. Apple is also not a biotech company, so I still did biotech there. So there's a couple of papers that are a bit out of Apple's usual expertise. So last year I did a quick sabbatical at Stanford and Genentech, working with Aviv Regev, my kind of like basically the research I was always looking up to. It was very fantastic working with her. So the work I'm presenting is still like somewhat part of my postdoc. I'm presenting part of my PhD, and then in particular I will present the very first work that is coming out of our lab. So we started at EPFL just 4 months ago. We are co-affiliated between the School of Life Sciences and the School of Computer Science, as well as have a courtesy appointment at the University Hospital in Geneva and the Precision Oncology unit there. So we have a very, very nice setup and this very similar ecosystem as what you have here in Heidelberg, where computer science and life sciences are basically all happening at the same place. So I guess I don't need to tell you that cell biology is complex. I'm also not so sure the audience, who is in the audience, but I assume that it's very biological. I'm still going to tell you what it makes it complex, because these are going to be a bit the motivating factors, like why we are working on what we are working on, what type of things do we need to build in order to solve the questions we want to ask. So biology is complex because the measurements we take, they're always indirect or they're obscure. Meaning, usually when I want to measure activity of a gene, I'm measuring transcriptomic counts, or if I want to understand like a phenotype of a cell, I often have to measure many different modalities. So we are a bit in a setting where most of our measurements are approximations of the processes that we really care about, and we need multiple different viewpoints or multiple different modalities in order to get a somewhat complete and good picture of what is actually happening. And so if any one of you have actually been in my talk that I gave an hour ago, that's identical. So then you should just straight into the room. So last but not least, so like often these modalities, we have a certain alignment, and this is very, very nice. So if we look at these new Xenium machines or Visio machines, they give us, of course, data of different modalities, different viewpoints of a biological systems that are aligned or somewhat aligned. But in the majority of the cases, this is not happening. So we have unaligned modalities, unaligned viewpoints of potentially the same biological systems, distinct measurements that we need to all incorporate to get a very good understanding of the underlying biological process. What are the driving factors of disease, things like that. And most importantly, and of course, this is not a complete list, but just like the three kind of, in my opinion, very important factors. Biological systems are inherently dynamic and driven by countless interactions. And even though this is the case, our measurements are often snapshots, meaning I take a measurement, I often have to destroy a cell, right? If I take a measurement of a cell. So even though I would like to have a readout of how a patient responds to a drug, predict the before and after of a perturbation, I destroy my individual system that I care about. I basically only get a measurement of either before or after. I only get a measurement of one modality. If I want to measure another one, I need to actually take a different sample, a different cell to take a measurement. So this entire dogma of machine learning, I have my X and my F of X, my output, and now I can learn my function F, something we are not provided with, and we need to come up with a better way of basically phrasing the problem, like writing down the problem, learning those architectures that overcomes this limitation. So we basically have non-time solved processes, and we need to learn and infer somehow these dynamic systems from snapshot data. So we see, and you're all aware, incredible success of those AI architectures in language and vision, like regimes where we have lots of data and very standardized data, and somewhat data that give us a fairly good representation of reality, right? I take a picture of this audience, even though, of course, it's a 2D out of a 3D world, it's a fairly good representation. I have a text that can perfectly describe some of my problem. So we are in settings where those architectures work very well. And this is, of course, A, because the problem is maybe a bit simpler, and B, because the architectures that have been developed over the last decade are very much tailored also to the data structures that we are working with there. So we have RNNs, we have CNNs, we have attention mechanisms, we have those specialized network architectures here, just a vision transformer or like a transformer used for text. They have been designed and tailored to text and image data. Same also the learning objectives that we are using, for example, the idea of GPT to predict the next token. That's a bit simplified. Of course, this works very well with language, but how would this look like for DNA? Just predicting the next base, it's not giving us a very good model or not a very good bias or learning instant to actually understand the structure, how DNA is formed and how it's read. We can read in several directions. Contexts are much longer. Relationships are not necessarily just sequential, depending on the organization of the DNA. And this is just one example. So what we were thinking of is what type of AI architectures do we need to really build those universal simulators for biology? And so this was a vision paper that has been published with Aviv, with Emma Lundberg, Yuri Leskowicz, Stephen Quake, and my two joint co-first authors, Yusuf and Yanai. And the idea is really, how do we build an AI virtual cell? And you shouldn't think of this as just a virtual cell, like one cell, and we simulate perfectly the behavior of a cell. Like it's more like a cell here as a unit of a biological system that, of course, operates within a tissue, ultimately an entire organism. And we need to be able to somewhat really simulate the behavior responses of those systems to perturbations, to changes, to adapt, like how it's adapting over the course of the development, to be able to understand or get a good understanding of those biological systems. How can this look like? So we know, of course, all that biology is incredibly hierarchical. We have those molecular components, and even these 3 scales are very, very coarse approximations. But we have, of course, molecular components. We have like a cellular readout. We have the tissue. And then, of course, it goes on and on and on. So we proposed the AI we see, AI virtual cell, like someone half a year later wrote a paper called AIDO, AI-driven organism. I forgot already. But so the idea here is really like somehow we have like models that are operating on the molecular scale. We have DNA language models, we have RNA language models, we have protein language models, or whatever model architecture you use. But they should ultimately feed into representations on the next scale. So we have cellular phenotypic readouts that we can connect to all the data that have been acquired on the molecular scale. And so it continues up until the tissue level. At the same time, we say that those systems are incredibly dynamic, meaning that they divide, they transform, things migrate. The entire kind of cells who form metastasis, they migrate through the body, through the human body, and they differentiate. And somewhat capturing those dynamics on top of those systems, which has basically all those hierarchical representations, is giving us some starting point to basically model cellular behavior over time. And so here we have basically operators or instruments that are sitting on top of those representations. And I mean, I think this is like one way where we can achieve a very first, or where we are able, like, which gives us an ability of combining all the data sets we've measured so far. One initial like versions of AI algorithms. I'm sure there are going to be proposals upon this on how to construct those systems that leverage all experimental measurements taken so far and things we can measure and put them into perspective. So how would this look like, right? So here we are basically moving away from this idea of like training an algorithm on a single data set to training an algorithm on all data sets. Here, just for one particular readout here, RNA sequencing, for example, we have those single cell foundation models that are trained on like by now millions and millions of data points, where we get some form of representation of the cell state. And such a representation of a cell state we could then use to make many different types of predictions and achieve many different types of functionalities. Meaning we can look at a mean of integrating many different data sources, right? Many different types of measurements. And I can always look at my one data set in connection of all the other data sets, or like kind of in relation of all the other data sets that have been measured. I'm able to incorporate many different species. We can look at spatial niches or cell states, identify somehow cell states that are new, that have a certain functional characteristic. But I can also look in particular at like how do those cells change upon perturbation, intrinsic, extrinsic in a developmental process. And last but not least, so this is mainly what my lab is interested in, we can now go into this active design mode. I can actively design a cell that has a certain property. And in particular, this is the last work that I'm going to also present now. We can find digital representations of our patients. So basically, I have a measurement, a clinic kind of obtained in the clinic, and I can embed it in a system that has been also seen many other different type of measurements. So even though all our clinical cohorts are pretty small and they might only have a couple of readouts, I can complement with whatever else has been measured, or other different clinical cohorts that have been kind of similar, but on this at the same time different. So we are getting away from this bottleneck of being limited to small clinical cohorts, because we are starting to have AI architectures that can operate across different clinical cohorts, even though they seem to be at the first place incomparable. So what I'm going to speak about today in the first place is like how to build such basically universal representations. I'm going to talk about a method that basically operates on the tissue level. And then I will talk about AI architectures that allow us to basically parameterize those neural operators, or virtual instruments, we call them, that allow us to basically predict a certain perturbation effect. So let's get started. So here we are working in a very particular setup. So within the Swiss Personalized Oncology Initiative that is running across many different hospitals in Bern, Basel, Zurich, Lausanne, and Geneva. And one of the main strategies of those is, or kind of like one of the main diagnostic tools usually is pathology slices. So we have H&E slices of a patient biopsy. On top, the Swiss Personalized Oncology Initiative also looks at spatial proteomics data. So they're collecting spatial proteomics data of patients. So here you have a patient. Does this actually have a pointer?
Oh wait, bottom.
So basically you have here a biopsy of a patient, and what you do is that you take basically images of those biopsy where you measure like the it's an antibody staining, so you measure actually the readout of many different type of protein markers. So so now you don't have normally in H&E you have an RGB valued image, like red, green, like it's a normal photo basically, where you see those like purple, pink stainings. Here we have a readout where we have like hundreds and hundreds of different channels, and every channel measures the intensity of a different protein. So how does this look like? So here we have like this patient biopsy. Let me just remove the sound. Here we have a patient biopsy, and you can see basically the intensity value of like a particular protein that has been measured, and we have actually hundreds and hundreds of different markers. So one experiment actually can even take up to 150. So spatial proteomics has just been announced method of the year by Nature Methods. And it's a quite it's gaining a popularity as really a core diagnostic tool in the clinic. What is very difficult is that we are dealing with small clinical cohorts, even though this is something run across Switzerland, that we have very different clinical labels. I mean, in Switzerland, we have this unique situation of having 4 languages or 3 main languages. Every hospital has a different type of annotation. We need to build algorithms that are able to do anyway those type of predictions independent of those heterogeneous labels. And last but not least, there's an O missing, we have a heterogeneity in the study design. Depending on my subject, I will have a different set of markers, right? Depending on the type of cancer. Okay, I lost my, oh, there it is. Depending on the type of cancer, there's different biomarkers that are important readouts to get a good idea of the molecular profile. Depending on the drug screen that has been run, we have different targets that we want to measure. So we will never see the same combination of channels ever, or usually we would assume sometimes we measure those 40, sometimes we measure this 50, sometimes we only measure these 5. How do we deal with this? Usually what people do in pathology, all those pathology foundation models that have been developed, they always have the same input. In standard image in RGB, always the same channels. None of these channels has even a meaning. These channels here are super high dimensional, right? We have to deal with hundreds of input channels and possibly different combination of those. How does it look like? So here we have, for example, cohort of 5 different data sets, and you can see the distribution of markers across those data sets. So sometimes they are present in several, sometimes they're only present in one. And this is the type of data integration and the type of challenge that we need to be able to deal with if we want to train one model across all those different patients. What we ultimately want to achieve is we want to create a virtual representation of my tissue, right? And this would then allow me to do, depending on what clinical label I'm interested in, or clinical task I'm interested in, or clinical labels I'm available, allows me to run many different types of downstream tasks. And here I care about downstream tasks that are happening on the actually even subcellular level or cellular level, because we have those spatial proteomics data, they are subcellular, have a subcellular resolution. I care about like certain niche properties. I might care about like something on the entire cell level. So this is just for biological discovery, understanding what contributed to a certain like disease phenotype and so on and so forth. But even more important, right, it allows me to do, I think it's just, sometimes it works and sometimes it doesn't.
With your finger.
Ah. Thanks so much. So we are also, of course, like all those like somehow clinical tasks that we care about. Relapse, what is the best treatment to choose, and what is a precise diagnostic cancer type, and so on and so forth. At the same time, we care about creating those virtual tissue databases across Switzerland, where we have basically all patients that we've ever measured represented in this database. And I can come with a new patient, compare them to all the cases that have historically been measured, and find basically similar patients understand what is their clinical history, what treatment has been successful, to use this really for clinical decision support. So this is the Virtuous platform that we constructed, and it really provides us now with a virtual tissue representation. We use this here in a setting where we train it on basically primary lung and breast cancer tissue. And so the ability we need to achieve now is that we can take a new patient, a new tissue, never seen before. For example, here we have primary melanoma cancer tissue that we can embed zero shot without fine-tuning or retraining the model to get a virtual tissue representation and now do all those different downstream tasks. So just to remind you, we need to be able to work with many different marker combinations. We need to work across different data sets. We need to be able to potentially even incorporate a new marker of a new clinical study that has never been measured before. We need to be able to deal with the dimensionality of channels that usual vision transformer and computer vision architectures cannot handle. And we need to do different predictions on different levels, cell, tissue, niche, and so on and so forth. How do we do this? What we developed is basically a new form of vision transformer that is designed to really excel on biological images, on multiplex imaging data. And for this, we first of all had to come up with a new tokenization system, similar as we have a tokenization system particularly designed for the language, and it should actually also matter if it's for German or English. There should be probably even a slight, like there's a less and a more optimal tokenization system. Here we need to think of a tokenization system for multiplex images. And of course, right, in RGB, nobody cares if it's R, if it's G, or if it's B. Like here we have hundreds of channels and they all have a biological meaning. They're all proteins that are connected, that have relationships, they interact, they take similar or dissimilar roles and functions within a cell. And we have this knowledge, so why not use it? So we basically have marker tokens that are based here on a protein language model like ESM that has been trained on the entire protein database. And we have a spatial token or image tokens, and we fuse them to basically get the tokenization system that we ultimately work with. And now we create this multi-scale architecture that has a cellular, a niche, and a tissue readout, and is thus able basically to do all those downstream tasks of all those different levels across of a tissue. How do we deal with the scale, and how do we deal with the fact that we ideally want to have some form of interpretability? We come up with a new attention mechanism that usually you have actually a fully connected attention. If you have hundreds of channels, this is not going to scale and this is not going to work. So we have a new attention that operates individually on the marker level across different markers. Which protein is important for each other? How can I leverage one protein information to make predictions on the next? And then we have a spatial attention. How do different tissue regions basically connect, matter, drive a certain development, and so on and so forth. We compare against the current state of the art by Recursion, a pharma company basically built on imaging data. And none of the current vision transformers actually is even applicable to multiplex images, to multiple channel inputs, and so on and so forth. Here, just using basically 10, 20, 40 markers, we stopped because afterwards it got too computationally expensive. You can see how the performance increase, like the runtime increases, like it's just incredibly costly because they are basically built on RGB images only. They straight away take Facebook's vision transformer and apply them on basically biological imaging data. This doesn't work. At the same time, what is even more interesting is the more channels I add, the more information I should get, right, of a biological problem. These vision transformers, they're usually completely channel agnostic because they just treat every channel individually. But these channels are not agnostic. They are important. One channel might be more important for the reconstruction of another one, and so on and so forth. It's all proteins that are operating within the same cell. We have architectures that are able to integrate this information, and you can see that the performance increases here on just the cancer type prediction of cell types, the more biological dimensions I add, the more channel marker, the more protein markers I add. Why is it so important to disentangle spatial and marker attention? Because it gives us two different ways of analyzing what the model has learned. All good?
I think I will put on the lights.
Okay, yeah. I think it's done. So on the one hand, right, here we have a biopsy where we just colored all the different cell types. We have here the immune cells, tumor cells, fibroblasts. And you can look at now basically spatial attention. Here, all the cells that have a black border, they are tumor cells. And you can see when we want to predict cancer type, and here we have the attention value, the model is looking at the tumor cells to make a prediction about the cancer type. This seems very logical to look at the distribution of tumor cells. For us, this is a very, very nice readout that the model actually looks at the relevant parts in the tissue. We not only have a spatial attention, we also have a marker attention. I can look at particular regions in the tissue, so dominated by tumor cells, dominated by immune cells, and look at what are the important biomarkers to make a certain prediction. And here you can see that in a tumor region, this is of course markers that are more like the kind of tagging or the kind of relevant for tumor cells versus here CD45 or CD20 in the immune cell, like heavily immune cell populated region of the tissue. Super important when we want to understand what are the important markers that we need for a measurement for a particular cancer type, for a particular patient, when we start to develop those minimal diagnostic tests, so that those spatial proteomics data are actually something feasible and scalable for the clinic. So why do we need zero-shot capabilities, right? Because now I want to take a new patient, possibly a new cancer type, and make those type of predictions. So here I take my melanoma sample, never seen any melanoma. We also do this for diabetic pancreas. And now I start to make basically those predictions. Here, biological discovery task on the level of cell types, tumor cell, T cells, trauma. And you can see that the performance zero-shot is almost as good as the performance on if we actually train on this data set. We can also look at tasks that are happening on the entire tissue. So for example, clinical diagnostic tasks such as predicting relapse, mutation stage, cancer stage. Here the prediction is done on the entire tissue. And then of course, it depends a bit, like sometimes like it's just performance is basically as good. Sometimes there's quite a heavy drop, of course, in performance. So here we perform against ResNet that has been actually developed for IMC type data. So the variety of diagnostics, so I show you two plots out of many. If you go to the preprint that's out since 5 days, you will see many of those different bar plots, many of different tasks, depending on the data set we look at. So why does zero-shot matter on the channel, on the marker dimension? Because what if I have a future study that looks at a different set of proteins, right? So here I train again on breast, lung, and breast and lung. We're looking at melanoma, which has a bit a different set of patients. So here you see the embedding of the protein language model. And for example, the other data sets, they've never seen MLANA or MLANA, not sure how people pronounce it. They also never see those like CCL8s, or at least like very different ones that are a bit further away. So now let's analyze like how does our performance, like how does our model do in integrating channels that it has never seen before. Let's start with the one that's somewhat well supported because it has seen many different proteins before. Because ultimately what we will feed in are those protein language models that are trained on the entire PDB. So here we have different masking strategies. So here we give the transformer a couple of pixels, and it might see for those pixels, or in particular for those pixels that are masked, it might see, other pixels of other markers. So it has a bit of an idea now on how this one pixel here correlates to other markers. And the marker attention that we have is able to basically pick this up on the fly. The protein language model embedding is able to put it in the right context. And you can see that the reconstruction ability is basically almost perfect between the original when trained on the dataset or when trained on the channel and when not trained on the channel. If I don't give any information, it's really truly a new image. It doesn't even see a single pixel. We can see that the performance is dropping quite a bit. But you can see the overall structure still picks up, but it's not able to get the right base level because it doesn't have any idea on how they are related. It's still very, very impressive that the overall structure is picked up, because now the only thing that operates on the fly is the protein language model and what is left from the marker attention. Of course, here you can see the performance, the moment here. So these are all the ones that are not in any other data set, sorted by how different they are compared to the protein language model embedding. And you can see that the performance overall is decreases. So this we call supported markers, this we call isolated markers. And then there's, of course, a clear drop in performance and ability to reconstruct. And of course, this is more a validation than a limit. It's of course a limitation, but it's also a validation because this is not a magician, right? So we need to feed it with the right information so that it can pick up those type of systems. What does it matter? Why does it matter? What are we currently doing? So it's not something hypothetical. The second last author these are people that are like that is the head of oncology in the Universitätsspital Zurich. I'm very, very closely working with basically the head of the precision oncology at HEG. Like we are really building this as a tool that it can be used in the molecular tumor board in Switzerland. So we are talking about this AI molecular tumor board. I can have a patient, I have a representation of my tissue, I query the virtual tissue database for cases, retrieve me cases that are very, very similar to what I've seen in the past. What we have here is two examples. So we have a patient, of course, this is something we might not know at the beginning. This is also a bit dumb labels, right? I can't put up and I can't publish all the kind of sensitive clinical information here. But you can see the 3 retrieved cases are somewhat very similar in the overall tissue structures, though they look also different. You can see it always finds the same adenocarcinoma. It looks at somehow similar grade, things like that. And so this is very nice. And so here I show you 2 examples. Of course, we quantify the retrieval statistics in the paper. So now I have basically a bit a historic context, and we collect those patients cumulative over the years in Switzerland, and we can somewhat put them into relation, of course, here only via their spatial proteomics, but this can be part of a very big ecosystem that also incorporates, of course, genomic information, histopathology, and so on and so forth. So how do we use the system now to do a bit more dynamic things, right? Like I want to predict the forecast how a patient would respond to a treatment, and then ultimately decide if this is a good treatment or not. So this is here like moving into the direction of really predicting behavior of individual cells. I have a cell and I have a measurement of a cell, actually also spatial proteomics data again. So where we have basically many different readouts as well as morphological properties. And now I want to predict how a cell changed from a healthy into a disease state, or in particular, how did the disease state transform of a cell upon perturbation with the drug? Meaning I don't only care, how the cell looks afterwards or how the overall population of cells look afterwards. No, I really care how every individual cell changed in their profile. What are the changes in expression or presence or abundance of proteins as well as morphological properties? What we always have is, you don't see, but it's light blue and gray cells, so very heterogeneous population of cells. And we, of course, want to be able to predict, how this sample of a tumor, for example, responds to a treatment. And as I said at the very beginning, these are unaligned data, right? The data samples we have, measurements are destructive. I'm talking about this just on the next slide, but I need to do this. I need to learn this from unaligned data. So just to describe a bit the setting, so we basically believe that those cells, they lie in a fairly high dimensional space. Here we have like 50 to 100 features, so it's actually not so high dimensional. And these are basically samples of a distribution of an unperturbed tumor. Samples because we need to here work on the level of distributions. Ultimately, I want to predict how those cells change upon perturbation of the drug. What I'm provided with are measurements of a tumor after treatment. So these are different cells because every time I measure a cell, I destroy it. So I basically split my distribution into 2, like my samples into 2. Some I treat, some I don't treat. And now I'm trying to understand how could this movement possibly look like? Which cell could possibly change to which phenotype, given what I measure after perturbation? Because exactly, I measure I need to destroy. I cannot measure the same cell before and after. I need to basically predict the overall distribution, change of distribution of those cell states over time. I can't do this on average because these cells are very heterogeneous. I very much care what is the subpopulation that is responsive, what is the subpopulation that is resistant, because this ultimately matters if I'm looking at a combination of treatment to understand, you know, how would the tumor grow after initial round of treatment. So somewhat I need to reconstruct these unaligned distributions. One way of doing so is to somewhat find a hypothetical pairing between what could be a cell here in this population of this measurement that could correspond to a cell state what I measured before. Ultimately, I don't care about this alignment. I want to do this out of sample. I want to give you a new population of cell and now predict how they would change. So I don't care about just like reconstructing those arrows, no. I want to have a map T that basically transforms my distribution of unperturbed states to a distribution of perturbed states. So we need to find an optimal map T that does this transformation. And I mean, one naive assumption, and this works well for the clinical observational studies that we use. By now, they are much more sophisticated that makes less of a strong assumption. But like one intuitive assumption is just look for a similar phenotype given some cost function. Because if I find a map that overall minimizes these misalignments, I probably have a good explainer of like a distribution change over time. And if I do this, I'll recover basically the core principle or the core principle behind something known as optimal transport, which all goes back to Gaspard Monge, friend of Napoleon, favorite of the French, still I think the founder of École Polytechnique has transformed French academics until today. And we're also not the first ones to think of this idea of using optimal transport. So Aviv Regev and Jeffrey Schiebinger have done this already in 2019, not in the neural network parameterized way, but then there's also work by Caroline Uhler and so on and so forth. So now we want to learn this map, and there's multiple ways, and I'm not going to bore you with the details because this is already like an old-fashioned principle because the machine learning community moves faster than you can blink. But so instead of reparameterizing this map, we look at one result that somehow says we can look at this as a potential, and in particular, like the gradient of this potential is giving us the map between unperturbed and perturbed, changes the objective. This is identical to the slide before, but now we basically only need to parameterize a function and take the gradient of it. And there's lots of work, and this is already, you can see I did this slide 2022. By now there's many, many more work. Also, we work more on this point of view from a diffusion model perspective now. Many, many different ways of doing so today. But it works very well because it gives us a map, this Monge map, to transform the distribution of unperturbed states into the perturbed states. So what I have during training is basically like samples of a patient before and after. So here, like these measurements and these discrete measurements, I can use this now to parameterize this map, this Monge map with the neural network, just what you saw on the slide before. We call this neural optimal transport. And this is what I wrote my PhD thesis on. And now after treatment, test time after training, we take this network and now we can predict how all those cells change and transform upon perturbation with the drug. And this is part of an entire series. So that was the final slide of my PhD defense last year. So this is not only a way, optimal transport does not only give us a way of parameterized maps, it has actually super beautiful and deep connections to dynamic systems theory and something known as gradient flows. Jordan Kinderlerer autoflows and have some work on that space. But much more powerful because it connects to all those diffusion generative models are the connections to control theory and diffusion models, flow matching, Schrödinger bridges, whatever name they take. And so if you're interested in how all those somehow theoretical concepts connect and how those different model architectures are related, we gave a talk last year on this 2023 at ICML. There was also recently another tutorial by Arnaud Doucet at DeepMind, who was speaking about this from a more diffusion point of view. So there's many, many materials by now that are providing an overview. Also, Aviv and I wrote a Nature Reviews method primer on all optimal transport in single cell and spatial omics for those who are interested. So what do we have now? We have basically a patient cohort, and we can train a generalized perturbation model. And now at test time, or like when I'm deploying this model, I have a new incoming patient and their cells. And now I can predict how they would respond to a particular treatment. And this was really designed for this tumor profiler consortium, which is a clinical cohort, observation clinical cohort that is running in Switzerland. Here we are working with 37 patients and a panel of 34 different drugs. And we use this for iPlatform, which is a spatial proteomics platform, but different from the spatial proteomics platform that we used in the first part of the talk. First part was Bernd Bodenmüller's IMC. Here, this is Lukas Pelkman's 4i platform. And this is two papers actually I'm presenting here, Nature Method from last year and Condor at Nerves from the year before. This one I did first, but Nature Method is just much slower than publishing in AI. So it's a very, very diverse cohort. You can see here all the patients here, they have all funny names. My favorite is Mudagok. He's somewhere here. I got to know them all, but only by this name, but you develop a strange relationship. And so then we have many different readouts like biopsy, subtypes, number of treatments they got before, because these are of course not standard of care patients. They have a treatment history, different driver mutations. And I mean, this is important because this is clinical features that we are also provided with. And they of course are not necessarily represented in the single cell phenotypic readouts we have, but important to condition on them and incorporate them because they of course are going to heavily impact the treatment response. And these are only a couple of them. There's many more confounding factors that we of course cannot account for. So now what we want to predict is basically how the cells change upon treatment. And I'm going to show you a couple of results. The paper is finally ready and is finally submitted. So here we see Panominostat, which is a drug, on a patient called Mogulib. And here is another drug for another patient. So basically these patients have never been seen during training. Now what we see here is the light blue curve for two different markers, and then we measure like many more. So these are two. So you will see like I can plot you hundreds and hundreds of those plots, and they are all in the paper. So we basically see just a couple of examples. Like you see that after treatment we have a movement from this light blue to this dark blue curve. So this distributional shift, of course, here this happens simultaneously in all the markers at the same time. And basically what we want to predict is perfectly capture this blue line. You can see that sometimes this is actually a bimodal response and only one part of the population is actually responding, and it's important that we basically capture this. The baselines, on the other hand, they just move everything to the left. So you see that both of those distributions have been shifted to the left. Our model captures the heterogeneous response that only a part of the population, actually a part of these patients' cells respond to the treatment and the other ones don't. It's of course very important to incorporate those these clinical features we have, because sometimes we have actually for dabrafenib a very different response depending on the driver mutation they have. For example, here for this particular patient that has this driver mutation, we actually see a response here from light blue to dark blue, and our model that has a conditional optimal transport that conditions on those meta information predicts a response, even though and the unconditioned models would not. Similar, sometimes we don't see a response where we would predict a response. Here, again in dabrafenib, like this patient then does not have the driver mutation, and so then it's always a bit off. Why does this all matter? So I think this is actually the most important slide of my PhD, why this was all important and all reasonable. So I'm very attached to this one bar here. It's my favorite bar. And so we don't only predict now the treated state, right? But we can really extract how every individual cell changed their features, because we can subtract the difference of the feature before and after. So we see the readout, the change in the readout. It's a clinical observation and study. So we actually have like somehow survival outcome or progression results. And so here the question we ask, like, is it an acute or is it a slow progression? And what you can see is that the predictions on the meta information and the control is not very informative. So the state of the disease before a patient came through this multiplex images gives us very little understanding of, the success of the treatment, of course. It's not just clear just from the beginning, before treatment has been given, what should be the answer. Even the treated cells itself, gives us a an improvement, but it's not like something that I'm very happy with. It really matters to understand how the patient responds. How do the cells respond to a treatment? That really gives us insight into the success of a treatment. And now we can basically go and predict throughout this entire drug panel the drug that would give us basically the highest level of progression. It also provides us a bit more insights into potential clinical associations. So here we run a kind of geometric statistical test on like different clinical information, as well as all the drug panels that we are working with. And so you can see that here for the treated cells, actually not even the predicted treated, there are actually way less clinical associations that pop up. And so, you know, often I heard the argument that, oh, why don't we just like screen all the patients and we have enough of those, like, you know, we just screen, it's getting cheaper and cheaper. No, it really, really matters to extract the change of cells over time. And many of those associations are also well-known facts, right? So for example, this connection here of paclitaxel and NRAS, we actually know that paclitaxel enhances microtubular stability, causing cell cycle arrest and cell death. And we know that patients with this driver mutation, they show very different activation of MAPK, so they have a different response to paclitaxel. So this will be out very soon. Finally finished the writing, and this also sums up what I wanted to say about. Where does this all lead? So there was a recent article in The Atlantic, I think a couple of days ago, like the AI virtual cell is a holy grail of science, it's getting closer, which is heavily citing and based on the vision paper I was introducing you earlier. So it's super exciting what is going on and how fast we are moving. The virtual tissue platform is a very initial somehow step within this like SPO initiative to take care, like to basically make use of those very diverse clinical studies. So I think I'm not sure what happened in 2012, so I'm not sure what we'll see in 2025, but I think we can all be very, very excited. So that's lots of work with many people, so I want to really take the time to do so. So on the one hand, there is my two PhD advisors, Andreas Krause and Marco Couturi, who are both not biologists, but with whom I have all done this optimal transport work, and they are fantastic. If anyone is looking for an advisor, I can highly recommend it. It was really, really fun, particularly being able to work with Anne Carpenter and Shantanu Singh for a couple of months doing my PhD at the Broad. Was very enriching. The work is really, like, especially the second part of the work is super close collaboration between Lukas Pelkmans and Gabriele Gut, the virtual tissue platform with Andreas Wicki, so and Oncology at the University Hospital in Zurich, and Gabriele Gut. And so they are, of course, my super close biological collaborators. It's really, really fun working with them. The last work has also been done in collaboration with Stefan, Hyung, and Bunne, and ETH. They are my two postdoc advisors who have basically written with me this AI virtual cell piece, in particular Yuri Leskevich and Aviv Regev. It was really, really fun. I very much recommend, and in particular my first co-first authors, Yusuf and Yanai. And so now all the work is happening with my team. So the two leading students of the virtual tissue platform, I mean, they arrived at EPFL 4 months ago, and they're doing a fantastic job. It's really, really fun working with such a dynamic team. They're super strong, and I'm very, very privileged to be able to work with them. And we are already actively hiring both on the PhD as well as on the master level, in case you're interested. Thanks so much.